So based on that research, I want you to see how a dendrite builds itself, how a dendrite changes. Just the tiny little dendrite, the little tiny, remember axons send, dendrites receive. And so the little tiny part of a neuron reaches out and grabs onto axonal fiber tracts and then links up, has these spines. And those spines are growing and receding all the time. And they are what are enabling new synapses to form. So to do anything, to think, to touch, to see, individual neurons have to organize their inputs. And one microscale brain organizational strategy is the spatial clustering of receptors on a dendritic spine. This kind of emphasizes to me how important it is that we work on very discrete skills with our patients, and how important we keep this idea of intense practice in mind. That discrete skills are important because they are what are going to build up, in part, and drive these individual neurons to organize their inputs. In this particular study, they looked at two kinds of novelty. Novelty, the first kind is called common novelty, where you visit a new beach, but you've seen many, many beaches. So you've seen lots of beaches and now you see a new beach. Versus distinct novelty, where this is the first time you've ever been to a beach. What are the different kinds of memory? And the interesting thing here is that when you are seeing something, a variation of something you know, then you have better semantic recall. So you remember better details and you can describe what you saw better. On the other hand, if it's novel, extremely novel, and distinct, you've never seen this before, then what happens is you have what's called episodic flashbulb memory. Now, as speech-language pathologists, we don't want episodic flashbulb memory. So what we want is enough repetition, but a little bit of variability each day so that they're building up their semantic system, their attention to detail, their knowledge of details about what we're working with. They're not going home with this vivid image of this brand new exciting thing they did that day. It's important to understand that when therapy is working, the child will become tired and so will you. The child will become frustrated and so will you. The child will become a little bit stymied sometimes. That's okay. That's part of the process of building the brain and changing the brain. If something's really easy for you, you're not changing the brain. We'll talk about that again later, but if you have a therapy session where the child doesn't get tired, where they aren't really being pushed, it might be good for carryover. It might be fine for just practicing, but it's not changing the brain. To change the brain, it does require the brain to wear itself out a little bit. I'll give you one example of a patient I had where the patient had recurrent utterances, and he was going, okay, 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 okay. And I used melodic intonation therapy with him, and I wanted to get him to be able to say the name of his girlfriend, whose name was Judy. So we did melodic intonation therapy, and he was able to say, my girl is Judy, my girl is Judy, my girl is Judy. And about 2 weeks later, his recurrent utterance, instead of okay, 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 became my girl, my girl, my girl, my girl. And so the neurologist said Marty was the etiology of his recurrent utterance. That was a maladaptive response. And it wasn't, what I didn't realize is by only doing the same phrase over and over with him and practicing that as opposed to doing a lot more variety, what I did is just created, literally as his brain was recovering from this illness, I just created another recurrent utterance without meaning to. And that enabled me to understand the importance of variety in therapy, flexibility in therapy. If you see maladaptive behaviors setting in, quickly moving off of them and finding another route or another kind of therapy to do with that person. And I was able to do that with him. That disappeared shortly after we started working on lots of other phrases, and I decided to avoid melodic intonation therapy with him for a while and use other repetitive language repetition approaches for a while and then go back to that perhaps later, and that worked. The problem with the lesion studies is that they focus primarily on the cortex. You can't really, with the lesion studies, it was difficult to see the fiber tracts involved. Even with the postmortem studies, it was hard to know how much of what you were looking at in terms of changes in the brain might have been post-injury or post-stroke. The research was a little bit vague and it was a little bit difficult to quantify. But the new data on how regions work simultaneously in networks that really started around the early 2000s up till the present. So for the last 18 years or so, we're starting to get a good sense of how these networks develop and become wired and change. And we can do that with functional imaging and tractography imaging. And that has dramatically altered our understanding of the brain organization itself and also the effects of experience on the brain. Now, the how of therapy, I'm going to spend more time with. It's a little bit more technical. There's a lot of research in here now. I've got a talk on that, but we're going to go into it with the new research in more detail. When you are driving reorganization, whether it's a child or an adult, what you are doing is changing neuromodulators. Neuromodulators are chemicals that enable the brain to rebuild itself. And we'll talk about what those chemicals are. They're naturally occurring. They're driven by behavior. So when we are doing therapy and we're manipulating behavior, we're also manipulating the neuromodulators. And it's interesting and fun to understand these. And this falls under really methodology or the how of therapy.